this is a good place to be, really good. So I'm honored to be here again and teaching tonight. Part of my joy and hobby is photography. So the scenic stuff you see is also the material that I've taken. This was recently taken in Hawaii, watching some birds fly. And let's work, okay. Tonight we're going to talk about change, transformation, and transmutation. We're going to look at what happens inside the body and what cells are like and how they respond to us for healing. We've got a friend with us tonight. This little bird was hanging out in Hawaii and is going to make several comments along the way about the work, so watch for that. These are astrocytes in the brain. They look like the cells in your brain. What's fascinating about them, and this came out of the most recent issue of Scientific American Mind, the May-June issue, which I received just before I got on the airplane, ran off a couple of scans to get this. 85% of your brain cells are not neurons. They're things called astrocytes, star cells. For years and years, and you will find in all the texts you read, that they're trashy cells. They just make a scaffolding that the neurons in the brain that do all the work hang on. But what's been recently discovered is they supply the links that make the blood supply to the neurons. They can produce their own neurotransmitters, so they're listening in and moving information throughout your brain all the time. If something happens to them and they get sick, they cause a number of serious problems. They're involved in epilepsy, they're involved in dementia, and this is how. This is what a normal astrocyte looks like, and it's kind of splashed out starry-wise. In dementia, the astrocyte loses its projections. It loses its ability to make the connections within your brain that help your brain work. It's also known that in depression, and this is a biopsy from a corpse from humans. This is not animal work. These are stained astrocytes in a normal brain. In depression, look how fewer there are. The follow-up work with this also went on that people treated with various kinds of things to raise depression actually repopulate the astrocytes. Obviously, those are a number of tandem studies to get that material. So here's a situation where we thought something simply supplied a scaffolding, and now we know that it's integral to the important work of our brains. I'm going to show you slides throughout the night just to let your mind rest. And so when one of these comes up, you know, just unfocus and relax for a minute. I was in Hawaii recently, and there during the tsunami, and this beach was completely remodeled by the force of the waves. No property was lost, no person was killed. But to see and experience something of the power of water in the ocean and of something that moves like a domino is a pretty awesome thing. Our Earth is an extraordinary place, as well you know. So I'm going to tuck in a few places here about the background that I show up with to talk to you, and because some of you have heard me and some of you haven't. So here's a little bit of it. That's me. Oh, not that long ago. At the electron microscope. An electron microscope weighs over a ton. It has an accelerating voltage of 100,000 volts that strips electrons off of a tungsten filament. Those electrons go shooting down a column, and all those knobs are for electromagnetic lenses that have to be very fine-tuned so that the beam comes right exactly where it needs to come through your sample, through the specimen. And then you see what's going on inside of a cell down in here. And underneath here is a whole packet of film. So you take pictures of what you're actually seeing. This machine allows you to magnify up to 1 million times. So you get to sit there with the lights down and the roughing pump going and the oil diffusion pump going and look at the secrets inside of cells. It's really a lot of fun. I also worked with scanning electron microscopy, and I'll show you that instrument later, but it looks at the surface of things. So some of the pictures I'll show you will be surface of cells, and some will be down inside of cells. I also worked with high-speed lasers. Here's an artist's depiction of a ruby laser. It's pumped until it emits laser light into this dye cell. Now, what we needed was a shutter. If you use a mechanical shutter, it's way too slow for the work we were doing. So the light goes into this dye cell, which is opaque. It just sits there and soaks up the light. Then all of a sudden, it's had enough, and it flashes totally clear in 5 nanoseconds. That's 10 to the minus 9th seconds. Faster than you can conceive. And out shoots a beam of red light that goes to your target. And then it closes down again. So this thing is called a Q-switched laser. Have you ever heard of a Q-switched laser? Anybody? Nobody's tried to have their tattoos removed? This is now used in dermatology. When I was working with it, it was an experimental laser that was being considered for working with fusion nuclear reactors. We found the nuclear reactors were far too unstable, so that research was dropped, but dermatologists have picked up this because it vaporizes pigment. So where there's a tattoo, and I had 5,000 of them all over me, I'm just kidding, where there is the pigment deposited from a tattoo, it will vaporize the pigment without burning the skin off. It's really quite an amazing thing.